Today, I will be teaching you the chapter Biomolecules. And one important thing to say before starting the class, that every week I will be uploading one biology chapter. Okay, sometimes 2 chapters, it depends, but most probably 1 chapter every week. Bio chapters, okay? So talking about biomolecules, we all know that we have to study about these biomolecules in 2 subjects. In 11th standard we have this in biology, and in 12th standard we have this in chemistry. You have to study this chapter in both classes and in complete different subjects in both classes, isn't it? In chemistry we are dealing with the chemical formulas, or I can say that chemistry aspects of this chapter. In this chapter, in the biology, we have to deal a vast topic related to this chapter. A vast topic is needed to be studied from this chapter. So without much introduction and stuff, I am going to directly start the chapter. Okay. Now, what do we know about biomolecules? Yeah, we know that biomolecules is a molecule. It is a different type of molecule that is present in what? In protoplasm. It is present in protoplasm. Okay, you know what is protoplasm, right? That is cytoplasm plus nucleus is what you call it as a protoplasm. So biomolecules are the molecules which are present in what? In this protoplasm. Now, is there only one type of biomolecule? No. There are many types of biomolecule, and all these biomolecules together form a cellular pool. Okay, about almost 40 types of biomolecules are present. All these 40 types of biomolecule together form a cellular pool in your protoplasm. Okay, now one more point. These biomolecules are also called as metabolites. Why do we call them as metabolites? Because they are used for the different metabolic activities that are taking place in our body. Whatever metabolic activities take place in our body, we use the help of our pyara dost, our biomolecules. That is the reason why we call them as metabolites. Now, we know that I told you about 40 different biomolecules are present. Same way, different elements are also present in the nature. This is just the introductory part. Okay, carbon is there, an element. Oxygen is there. Nitrogen is there. Hydrogen is there. Do you know how the order goes? Yes or no? The order goes in this way: oxygen first, carbon, then nitrogen, and then your hydrogen. This order is seen in your body. When it comes to Earth's crust, how is it present? Means first you have oxygen itself. Oxygen is abundant everywhere, whether it's in human body or whether it's in Earth's crust. Oxygen is the abundant. Okay, oxygen. After that you have hydrogen. Oxygen is almost 46 point something percentage, and hydrogen is 0.14 percentage. Then you have carbon, which is 0.03 percentage, and then you have nitrogen, which is just in a negligible amount, because when carbon itself is in 0.03 percentage, we know that then this nitrogen will be in a negligible amount. In talking about your human body, oxygen is, I said, whether it's human body or Earth's crust, oxygen is the most abundant one. That is almost 65% of oxygen is present in human body. Then carbon dioxide is 18.50%. I think this percentage was studied in your chapter breathing and exchange of gases. Okay, not all, some percentage, because in there we only need oxygen and carbon dioxide. Okay, nitrogen is 3.30% and hydrogen is 0.50%. This is how the percentage goes. Okay. Now, let us just get into the chapter. Okay, let us just get into the chapter. That is Biomolecules. Biomolecules can be differentiated with the help of two tests. One is Ash test and another one is acid filter test. Ash test and acid filter test. Now, what is Ash test? How do you classify based on Ash test? Based on Ash test, you classify biomolecules as organic and inorganic. Based on acid filter test, you classify biomolecules as micromolecules and macromolecules. Now, talking about acid filter test, the acid which you use here is none other than trichloroacetic acid. Trichloroacetic acid. Cl3COOH. The formula itself you can know, trichloro, 3-chlorine, acetic acid means COOH. Okay, so the acid which you use here is trichloroacetic acid. Now you have a separate thing to discuss. That is, what is micro and what is macro. It is a separate thing which you have to discuss. Okay, so talking about micro and macro, just copy paste them. Okay. Micro and macro. Micro means small, macro means big. We all know that. So what's the special thing to learn in this? The thing is, what you said or what we know is an important point regarding micro and macro molecules. Micromolecules means their molecular weight is less. How much is it less? It is less than 1000 dm. So what will be here? Their molecular weight will be more than 1000 dm. One is small, one is big. That is the main thing which we know. One is small and one is big. So then what is the special thing? The thing is, microbiomolecules are filtrate. Do you know what is the meaning of filtrate? And this one will be retentate. Do you know what is the meaning of retentate? Yeah. Filtrate means what? Which can pass through the filter. You know that this micro and macro molecules are separated using the acid filter test. So acid filter test. So this acid is being used here. So it is a liquid substance and it will definitely pass through the filter. So a substance which is of less weight will mix with the acid and will pass through the filter. Which is less weight? Micromolecule is less weight. So it will pass through the filter. It's definite. That's the thing. That's why it is known as filtrate. Now what is retentate? Which retentates back. Which stays back. That is acid insoluble. This is acid soluble and this is acid insoluble. Of course. That's why we are learning all these, isn't it? Acid soluble and acid insoluble. So retentate means acid insoluble. Now what are the examples? Just think. I'll give you 2-3 examples and you just think whether it will be micro or macro. Just think of DNA or RNA or polysaccharide or proteins. Okay. DNA, when we think itself in our human body, we study thousands of DNA which is made up of many chromosomes and genes and everything composed together. RNA also single stranded. Then polysaccharide means many sugar monosaccharides combined together. In biomolecules in chemistry we learned it. Monosaccharides combined together, more than 1000 or 100,000 monosaccharides when combined together form this polysaccharides. Proteins, many amino acids combined together and form proteins. So combination of things is taking place here. So what will it be? Of course, there is no doubt in it. That is, it will be macromolecules. It will be macromolecules. All of them will be macromolecules. Okay, DNA, RNA, poly, protein, everything is a large substances and they will be macromolecules. Pakka, that we learned. Now, Now micromolecules. Protein is made up of amino acid, which is a micromolecule. Like that, you will remember it, right? Polysaccharide is made up of monosaccharide, which is internal sugar. Of course, DNA, RNA made up of nucleotides, which is a micromolecule. Then water and all chintu substance, water, vitamins, very chintu. Then minerals, etc. minerals, etc. See, everything, these all come under your micro macro particles, your micro macro particles. Okay, see, okay. Now, an exceptional case which you have to learn here is, an exceptional case is lipids. We all know about lipids and we learned about lipids. This lipids is a micromolecule. It is a micromolecule, that also we know. Why is it a micromolecule? Because its size is less than 800. Less than 1000 itself, less than 1000 itself comes under micro. So this is less than 800. So it is definitely a micro. But what is the exception here? Because lipids is just like amino acid or vitamin. It is a micromolecule. Its size is less than 800 dalton. What is the difference? Difference is, even though it is a micromolecule, it is present as retentate. That is, it is acid insoluble. It is present as acid insoluble. Okay. So what is the exception here? Lipids, even though they are micromolecules and even though their size is less than 800 dalton, they retentate. Okay, they retentate, and thus they are an acid insoluble compound. Now, why do they retentate, or why are they called as acid insoluble compound? I will teach you. Okay, when we are dealing with lipids separately, I will be teaching you what is the reason why they are called as retentate. Okay. Now, next is, you have to know that in protoplasm, mostly water is present. We know everywhere in our body itself. Main is water. The main thing which is present in our body is water. It's biomolecule. Okay, so the most abundant biomolecule, or the most composited biomolecule in our cell is water. Water is the major composition. That is almost 70 to 90% of it you have water. Okay. Then you have protein, which is almost 10 to 15% of protein is needed or is present in your cell, in your protoplasm. Okay, anything. Then lipids are just 2%, just 2% lipids. Nucleic acids, main thing, but it is just 5 to 7%, not up to proteins also, 5 to 7%. Then you have carbohydrates. Every day you are eating rice and all. Then what is the amount of carbohydrates in your cell? Just 3%. And then rest inorganic ions, just 1%. Just remember these data, no need to buy heart and all, just remember it. It is water, almost 70 to 90% water is present. Protein is 10 to 15%. Then you have nucleic acid, which is 5 to 7%. Then you have carbohydrates, 3%. Lipids, just 2%. And inorganic ions, just 1%. So once again, water 70 to 90%, protein 10 to 15%, lipids 2%, nucleic acid 5 to 7%, carbohydrates 3%. Even though you eat rice all day, it's just 3%, and inorganic ions 1%. Okay, now next is metabolites. Okay, metabolites means the biomolecules, okay, which are important or which are involved in our growth and development are known as primary metabolites. So metabolites are also of two types. Primary metabolites and secondary metabolites. So what is primary metabolites? The metabolites which are important for your growth and development is what you call as primary metabolites. And what is secondary metabolites then? Secondary metabolites means that there are some biomolecules which do not have any major role in metabolism of the producer. Okay, no major role in the metabolism. They are just useful to human welfare. Okay, like this example I will just say, morphine and all, morphine, then rubber or gums. All these are not important for your metabolic processes, means growth, development and all. That is just primary metabolites which are important for your growth and development. For example, your amino acids, lipids and all. They are important for growth and development, and thus they come under the category primary metabolites. But secondary metabolites means they are not present or they are not involved in your metabolic activities. They are just important for human welfare. Rubber it is for us. Gums, you chew mostly, isn't it? Morphine, used for making medicines, or lemongrass oil, gingerly oil, or you can say cellulose, rubber, gums and all come under polymeric substances, or like some toxins or some terpenoids like diterpenes and all. Then some oils, I said lemongrass oil, gingerly oil. Then some pigments, anthocyanin, carotenoids, anthocyanin, color, isn't it? Carotenoids also, orange colored pigments present in plants. So mostly all these things, when I told you about all these things, you have one thing common stripe. So you have one thing most common struck in your mind. What is the thing? Yeah, that is, whatever examples I told you in the secondary metabolites, all of them are coming from plants. Are you producing anything? No. Are any other animals producing anything? No. So the secondary metabolites, the main thing is they are absent in animals. That is the main thing. Absent in animals. Sorry, just got an important call. That's why I'm so sorry. Okay, so morphine, rubber, gums, etc. Okay, so all these things, you know that they are given by animals. Plants, and they are absent in animals. And all these things are for our welfare. And these things are put into the category of secondary metabolites. So is there any doubt regarding primary and secondary metabolites? I hope nothing. If you have any doubt, just put it in the comment box. I will be clarifying it. Okay. So metabolites, there are some metabolites that are involved in growth and development and are known as primary metabolites. Example, amino acids, lipids, vitamins, etc. Some biomolecules have no major role in metabolism of the producer. Okay, we are the producers, and do they have any, for example, okay, in our case, we are the producers. In animals' case, if we are talking about cat is the producer. Okay, and does the secondary metabolite have any role in the metabolism of the producer? No, and such biomolecules are known as secondary metabolites. They are generally present in plants and absent in animals. And most of the secondary metabolites are useful to human welfare. Okay, some pigments are seen in this, anthocyanin, carotenoids. Some drugs are seen, reserpine, curcumin, windblasting. Some oils are seen, lemongrass oil, gingerly oil, etc. Some toxins, resin, abrin. Some polymeric substances like rubber, gums, cellulose, etc. Or some lectins like concanavalin A. Okay. Now, this is about metabolites. Now next I'm going to, so today most probably in this one class, okay, I will be uploading this chapter as 2 parts. So in this first part I will be most probably covering till let me check till proteins. That is, I will be covering proteins also, lipids, nucleic acid and enzymes will be taken in the next part. Or do one thing, I will just complete this nucleic acid, which is just small part. I will be completing nucleic acid also, and if the time allows, then today evening itself the second part will also be uploaded. Okay, second part will also be most probably uploaded by evening. In first part I will be completing up to lipids. Lipids will also be taken. Then you have just nucleic acid and enzymes left. Okay, so fast. So carbohydrates. Yeah, come on. Let me just make myself a little energetic so I can continue the class. You just have some water and come, okay? So, okay. So, if you want, just pause the video and take a 2-minute break and come fast. Yeah. Willing to continue till I finish carbohydrates. After carbohydrates, I will be giving a 2 minutes break. Okay. So, first let me just finish carbohydrates. Okay. Fine. Carbohydrates. What is the formula of carbohydrates? Can you just tell me what is the formula of carbohydrates? Like not the general. I just want the general formula. Not a general formula. Just give me the formula of any one carbohydrate, like glucose, sucrose, or anything. Glucose, okay. Glucose, okay. So you just give me the formula. C6H12O6. So in a carbohydrate, seeing the formula itself, okay, glucose. One more, just give me the formula of one more carbohydrate, sucrose. Okay. C11. Oh, I'm sorry, it's just getting cut sometimes. Okay. Yeah. So sucrose is C12H22O11, isn't it? Seeing this formula, we can make out one thing, that carbohydrates are made up of three elements. What are these three elements? Carbon, hydrogen and oxygen. Carbon, hydrogen, oxygen. So with these three things is what your carbohydrates is made up of. And this one is usually they are combined in the ratio. C, H and O are combined in the ratio 1:2:1. See, 1:2:1. Here it is different because it's 12, it's 11. It can be 1:2 and it's 22. It's 24, and if here it's 12, we can say it was 1:2:1. This is just an exceptional case, but in most of the cases, this glucose means this carbohydrate will be combined. The carbon, hydrogen, and oxygen will be combined in the ratio 1:2:1. Okay. Now, carbohydrates are the main energy providers of our living system. Okay, and these carbohydrates are divided into three, that is monosaccharides, oligosaccharides, this one we studied in chemistry, and polysaccharides. This is a point which we studied in chemistry, mono, oligo, polysaccharides. Now what is monosaccharides? Yeah, we know monosaccharides means they are the simplest sugar. Now can we divide them? No, they cannot be hydrolyzed, or they cannot be divided into further products. Okay, they are the simplest sugar. That is what you call it as monosaccharides. Okay, simple sugars which cannot hydrolyze into other sugar molecules. That is what you call as monosaccharides. Now, as an example of monosaccharides, yeah, glucose. Can you now just separate them into any other? Can you just divide into any other carbohydrates? No, glucose is glucose. Same applies for fructose, ribose, glyceraldehyde or dihydroxyacetone, ribulose, etc. Everything, okay. Ribulose, glyceraldehyde, GCA, aldehyde glyceraldehyde. Okay. So all these come under monosaccharides. This glucose and this fructose are hexose sugar, means 6 carbons are present. So let's see, glucose 6 carbons. Ribose and ribulose means pentose. Both of them are pentose sugars. 5 carbons are present. GCA and dihydroxyacetone is triose. Just an example. Okay, I'm just mentioning all these things. Now, if this is what you mean by monosaccharides, then what will be oligosaccharides? Yeah, oligosaccharides means that is from 2 monosaccharides, from 2 to 8, 2 to 10, like between that. Okay, if 2 monosaccharides are combined together, you call them as disaccharides. These disaccharides come under the category of oligosaccharides. Okay, that is, oligosaccharides itself is divided into categories, that is disaccharides, trisaccharides, tetrasaccharides, etc. Okay, yeah, you only have to study about disaccharides. Okay, only about disaccharides, but still I'm just saying oligosaccharides are divided into disaccharide, trisaccharide and tetrasaccharide. Now, disaccharide means what? 2 monosaccharides. Tri means 3 monosaccharides combined together. Tetra means 4 monosaccharides combined together. So 2 monosaccharides combined together are what you call it as disaccharides, and they are always linked by glycosidic bond. They are always linked by glycosidic bond. See? Okay, and there is a C carbon-carbon bond formed. C bond is formed, and this bond is joined together by glycosidic bond. So glycosidic bond, carbon-carbon is joined together by glycosidic bond. Now there are examples of many disaccharides. That is, you can have maltose as an example, because when maltose is hydrolyzed, you get what? 2-glucose, which is a monosaccharide. Hope you understood what is a disaccharide. With example you can understand. Disaccharide maltose is an example of a disaccharide. When you hydrolyze this disaccharide, you got 2 monosaccharide, 2 units of monosaccharide, that is glucose. Is there any compulsion, or is there any is it compulsory that we get only same monosaccharide? No. Okay, just take the example of sucrose. Take the example of sucrose. When you hydrolyze it, you get what? Glucose and fructose. You get glucose and fructose. 1 unit of glucose and 1 unit of fructose. Okay, so this is what we know about disaccharide. Now one more example of disaccharide is this one, lactose. Lactose, you get glucose, 1 unit of glucose and 1 unit of galactose. Okay, both of them are. Galactose is also monosaccharide. Okay, but can you separate this glucose now? No. Can you separate this galactose now? No. Can you separate fructose now? No. So that is what is meant by monosaccharide, disaccharide, etc. So this is what you have to study about disaccharide. And one more thing, in maltose, these glucose units are there. We said that this one are connected with the help of, that is 2 monosaccharides are connected together with the help of glycosidic bond. Okay, here in case of maltose, it is also connected by glycosidic linkage, but between C1 of alpha glucose and C4 of another alpha glucose. So you have a alpha 1,4 glycosidic linkage. Okay, that you studied in chemistry. I hope that you studied it in chemistry. That is, C1 of alpha glucose connects with the glycosidic linkage to C4 of another alpha glucose. You have C6, isn't it? 6 carbons are there. Like this in glucose, you have 6 carbons. This structure you studied. So this structure. Many helping structures are there, but mainly 6 carbon structure you studied. 4, 5 and 6. So within this alpha glucose's first carbon, and you have another carbon also in this 4th. So there is a linkage happening between these two, and this results to your what? Maltose. Hope you understood what I meant. Okay. Let's do this. Okay. Now, lactose is glucose and galactose. Sucrose is glucose and fructose. Now the sucrose is a non-reducing sugar. It is a non-reducing sugar. Just remember that point. The reason and all you studied in chemistry, I am not repeating it here. It is a non-reducing sugar, and therefore it is the main transporting form of carbohydrate in plants. So carbohydrate are mainly in plants. Carbohydrates are mainly transferred in the form of sucrose, because it is a non-reducing sugar. Okay, in plants, carbohydrate is mainly transferred in the form of sucrose because it is a non-reducing sugar. Hope you got this also. Fine. So, this is what you have to learn about disaccharide. Now, I will just teach you about glucose and fructose in a more detailed way. Okay, nothing more detailing, just introduction should be given about both, right? Monosaccharide, or they will just feel lost. Glucose. Okay, glucose is a hexose aldose reducing sugar, and it is also known as grape sugar, blood sugar, etc. Okay, so glucose is mostly, we call it as grape sugar. Okay, and it is a hexose made up of 6 carbon. It is an aldose because an aldehyde group is present in it, and it is a reducing sugar. These are the characters of glucose. Just remember these. It is a hexose because 6 carbon. Aldose because it has a CHO group, a aldehyde group. It is a reducing sugar. Okay. Now, do you need the structure of it? I'll just draw it. Sorry, what did I draw? Okay. So this is the structure. Like this, isn't it? It goes like this. Okay. Now, here you have. Just change the color so that you just don't get confused. Here you have CH2OH. Here you have HOH, OH. In second carbon there is a difference, HOH and OH and H. Okay, so this is the structure of glucose. That is, this third carbon has a difference, isn't it? First, second, third, fourth, fifth, sixth. Third carbon has a difference. OH is goes below. Here the H is below. H is present in its below position, and OH. You understood it. Okay. This is the structure. Now, next thing we have to learn is about fructose. Next is fructose. So, what is fructose? It is also a hexose 6 carbon, but it is a ketose because a ketone group is present. It is also a reducing sugar, and it is the sweetest non-sugar. Sometimes you would think that sucrose is the sweetest sugar, but no, the sweetest sugar is fructose, and it is present mainly in fruits, so it is known as fruit sugar. Glucose is present in blood, and that's why we learn in hospitals and all we hear, right? In glucose is present, just check the level of glucose in blood, so they are also known as blood sugar. Okay. Fructose is fruit sugar. Okay, so fructose is a hexose ketose reducing sugar, and it is also fruit sugar. And the thing about fructose is in male it is present in some glitch happened. Okay, so it is present in seminal plasma in male. Okay, so it is present in seminal plasma. Okay, so disaccharides, you studied about monosaccharides, disaccharides. Disaccharides example are maltose, sucrose, lactose. Maltose is made up of glucose, 2 units of glucose, each connected together by C1C4 alpha linkage. And next you have sucrose, which is made up of 1 unit of glucose and another unit of fructose. This is a non-reducing sugar. Therefore, in plants, a carbohydrate is mainly transferred in the form of sucrose. Then you have lactose, which is made up of 1 unit of galactose and 1 unit of glucose. Okay. Now see, non-reducing sugar. Sucrose is non-reducing sugar. Okay. Now next you have about monosaccharide, that is the glucose, which is also known as grape sugar or blood sugar, and it is a hexose, it is an aldose, it is a reducing sugar. The structure, Haworth structure. Okay, you call it this as Haworth structure. This is a Haworth structure. And then you have fructose, which is a hexose ketose reducing sugar, and it is also a fruit sugar. It is also present in seminal plasma in male. In seminal plasma, the enzymes of seminal plasma. The seminal plasma is rich in fructose, and all that was there to learn in human reproduction chapter, right? Now, mono is completed, di is completed. Next is polysaccharides. I'll just complete it fast, so that within 1 hour I just thought of completing already almost 35-37 minutes are over. So I just thought of completing the chapter within 1.5 hours. I'm not sure, but I'll just try my maximum. Okay. Part 1 to be completed, not the chapter. I'm sorry. Part 1 to be completed within 1.5 hours. After this biology, I have decided to take the chapter Plant Kingdom. Okay. Sometimes I will also be taking physics and chemistry. I'm not sure about it. Most probably chemistry will be taking physics. I'm not sure much. But still, I'll just try my maximum. Okay. So polysaccharides. Polysaccharides, is it a micro or is it macro? I thought it is a macromolecule. It is a macromolecule, isn't it? So polysaccharides are macromolecule. When you talk about protein also, just use the word macromolecule. When you talk about what next, DNA, RNA also, use the word macromolecule or macromolecule. Okay. Oh, and one more thing which I wanted to correct was, I'm sorry, I made a mistake here. It is in macromolecules, right? I told that macromolecules, I said that which is more than 1000 elements, which is wrong. Only when I saw it, I wanted to change it, but I forgot. It is more than 10,000 Dalton. Okay, this one is less than 1000, and this is looking like 100 or something. Oh. So 1000. So it is less than 1000 Dalton as micromolecule, and more than 10,000 Dalton is macromolecule. If you want these notes, just comment it. I will be giving it. It's publishing it. Now talking about polysaccharides. Polysaccharide means it is a macromolecule formed by the polymerization of monosaccharides. Isn't it? Many monosaccharides being combined together form polysaccharides. So examples are cellulose, chitin, starch, glycogen, and all examples of these polysaccharides. Okay, that is many units of monosaccharides comes together to form this polysaccharide. In a polysaccharide state, first you just say glycogen. The right end is called reducing sugar or the reducing end, and the left end is called as non-reducing end. Okay. In glycogen, see, I will post a picture related to glycogen here so that you just get an idea. Okay. Wait 1 minute, I will be posting here. So you see, this is the structure of glycogen. So in this glycogen, you see this is given in your NCERT. Okay, it is an NCERT based structure of glycogen. So in this right end is called the reducing end, which is the right end. This is the right end, isn't it? So this right end is known as reducing end, and this left end is what you call it as non-reducing end, NRE. Okay, so right end is reducing end and left end is non-reducing end. Just remember that in a polysaccharide, in this disaccharides and all, we call the whole disaccharide as reducing or non-reducing. Same applies for monosaccharide. But in a polysaccharide, the right end is reducing end and the left end is non-reducing end. Now, types of polysaccharide based on mono units. Homopolysaccharide, which consists of a single monosaccharide unit. That is, for example, cellulose. Just take the example of our most common brother, cellulose. So when you talk about cellulose, we know that it is a unbranched homopolymer. It is a homopolysaccharide, I said. So it is a homopolymer of what? Of glucose. It is made up of only glucose. You do not have any other monosaccharide present in cellulose. The cellulose is only present in glucose. And cellulose, it is an unbranched homopolymer, because you see this is the branched homopolymer. See, can you see the branching and all? See, branching and all. Can you see this? So this is the branched homopolymer of glucose. Cellulose is the unbranched homopolymer of glucose. It is not branched. Okay, and it is the most abundant polysaccharide. Okay, most abundant, not most abundant element. Okay, most abundant polysaccharide is cellulose. The most seen polysaccharide in the universe is, or in the earth is, cellulose. And we know that cellulose is present in the cell wall of plants and all. We studied many things in green colored plants. You can see cellulose. Many facts were studied about cellulose, and this is the extra fact. It is a homopolymer of glucose, unbranched homopolymer of glucose. It is the most abundant polysaccharide. Okay, paper made from plant pulp and cotton fibers are everything is cellulose. Now it is a structural and supporting. An important point, very important point to be noted: structural and supporting polysaccharide. Structural and supporting polysaccharide is cellulose. It is a structural and supporting polysaccharide, and it shows a beta 1,4 glycosidic linkage. It shows beta. Alpha 1,4 glycosidic linkage was shown by maltose. Maltose, isn't it? Maltose has 2 glucose units which are joined together by 1,4 glycosidic linkage, and the 2 glucose units are alpha glucose. Here also you have more than 2 glucose units, and it is unbranched, and also it is beta glucose. Here also you can see the linkages between C1 and C4 of 2 glucose units, but the glucose unit is beta glucose. You know what is beta glucose, isn't it? In your chemistry you learned about it. Okay, so these are some important points. You just remember in such way. Maltose is a disaccharide. It shows alpha 1,4 glycosidic linkage. So if a disaccharide is showing alpha glycosidic linkage, then some other saccharide will be showing beta glycosidic linkage, and that saccharide is polysaccharide, that is cellulose. Okay, cellulose is showing that beta 1,4 glycosidic linkage, and it is a structural and supporting polysaccharide. Next example of this homopolysaccharide. So I told that this polysaccharide are of 2 types, homo and hetero. I think you understood what is the meaning of homo. Homo, cellulose is example of homo, made up of only 1 type of monosaccharide unit. Okay, glycogen is also an example of this one. Glycogen is also an example of homopolysaccharide. The thing is that just the differences, I just erase it and I will show you. The differences, see, can you see the branching? The branching can be clearly seen. See, branching. This branching is what makes it different from cellulose. It is a branched homopolymer. It is a branched homopolymer. Branched homopolymer is glycogen of glucose, and it shows what 1,4 glycosidic linkage. See, 1,4 between these, 1,4 between these, and then here you can see the 1,6 glycosidic linkage also. Okay, so you can see 2 types of linkages are seen here. Two types of linkages are seen here, 1,6 glycosidic linkage and 1,4 glycosidic linkage. Okay, alpha, both are alpha. Now if the cellulose was the functional and structural polysaccharide, this is a storage Okay, so glycogen, it is a storage polysaccharide. Okay, glycogen is a storage polysaccharide and it is a branched homopolymer of glucose. Homopolymer and a heteropolymer. Now, example of a heteropolysaccharide is hemicellulose. Hemicellulose is an example of a heteropolysaccharide. Wow, okay, you don't have to study in detail of this hemicellulose. Homopolysaccharide and heteropolysaccharide. Cellulose is homopolymer of glucose, unbranched, most abundant. I'm just revising it, okay? Unbranched homopolymer of glucose is cellulose. It is the most abundant polysaccharide, and it is a structural and supporting polysaccharide, and the linkage is beta 1,4 glycosidic linkage. It is seen between C1 and C4 carbon of beta glucose. The linkage of cellulose is seen between C1 and C4 of beta glucose. Now you have alpha 1,4 glycosidic linkage, that is for maltose. Okay. Now the next one was glycogen. It is a branched homopolymer of glucose. Two linkages can be seen: 1,4 alpha linkage and 1,6 glycosidic linkage. Alpha. It is a storage polysaccharide in animals. Okay, in animals it is. In plants it is starch. In animals, the storage polysaccharide is glycogen. Now next is chitin. We're talking about chitin, you know. In fungi and all, their cell wall is made up of this chitin. It is an example. This chitin is an example of a polysaccharide. It is the second most abundant polysaccharide. First is cellulose. Chitin is the second most abundant polysaccharide. It is also a structure and supporting polysaccharide. And supporting means it is formed in, it is seen in the cell wall, which is a supporting structure, isn't it? Structural and supporting structure in organisms. So chitin is also seen in the cell wall of arthropods and all. In the exoskeleton of arthropods and fungus and all. You can see chitin. Okay. You can see chitin in the exoskeleton of arthropods or in the cell wall of fungus. It's the second most abundant polysaccharide. Okay, it is also an unbranched polymer. It is also an unbranched homopolymer itself. What all I am teaching here means been explained, but the explanations whose all polysaccharides examples or explanations are given. They are all homopolymer. Only one heteropolymer I taught you, that is hemicellulose. Okay, so unbranched homopolymer of, is it of glucose? No, unbranched homopolymer of glucose is cellulose. Unbranched homopolymer of N-acetyl glucosamine, because you know once question was asked, not from this chapter, from I think from first chapter, first or second chapter, you have to study about all these fungus and one chapter is there, no? In that chapter, one question was asked in a mock test that the cell wall of fungus was made by combining many acetyl and acetyl glucosamine. Many of them just gave that one assertion was there and another one was reason. The assertion is correct and reason was wrong, but the answer was both are correct, but it was not the correct explanation, but both are correct was the answer. The reason why everyone made mistake is because everyone will think that fungus cell wall is made up of chitin. So N-acetyl glucosamine, nothing. So every chapters are connected, my dears. Every chapter is interconnected. So when you are learning about fungus cell wall in that chapter, I think in your second chapter, biological classification, you have that. So when you are studying in that chapter, you have to remember that this chitin is the homopolymer of N-acetyl glucosamine. Okay, homopolymer, that is many acetyl glucose amine when combined together form this chitin. Okay, polymer means polymer and monomer, that also everyone will get confused. Sometimes it is asked that this fungus is made by many fungus cell wall is made by interconnecting many chitin. It's wrong. When many N-acetyl glucosamine interconnects, that's why this chitin is the homopolymer of your N-glucosamine. It is the homopolymer. N-acetyl glucosamine is the monomer of chitin. Two ways the questions can be asked. N-acetyl glucosamine is the monomer of chitin. Chitin is made up of this N-acetyl glucosamine. Okay. Many chitin together don't combine together to form this N-acetyl glucosamine. Many N-acetyl glucosamine combines to form your chitin. Let's just remember it, and just remember that it is an unbranched homopolymer. Okay. Now, what is the next thing you want to learn is starch. Starch is also a homopolymer. Starch is also homopolymer of glucose. It is not mentioned that whether it is branched or unbranched. It is just homopolymer of glucose, because starch are of two types: amylose and amylopectin. Amylose and amylopectin. Talking about amylose, it is the unbranched one. Unbranched glucose chain present in starch. Amylopectin is branched chain. And you know what? The secondary form of amylose, it can change this iodine to the blue color. In experiments and all, starch is used. To test for starch, we use iodine, and this changes it to deep blue color. That we learn. This amylose, the secondary form of this amylose, is the one which is changing your iodine to deep blue color. In starch, you can see both. Amylose is also seen, amylopectin is also seen. Amylose is the unbranched glucose chain, and amylopectin is the branched glucose chain. Okay, it is a homopolymer of glucose. It is also made up of only glucose. Okay, it is also a storage polysaccharide in plants. Okay, storage polysaccharide in plants. In animals, it is glycogen. Storage polysaccharide in plants is starch. So, is that also okay? Now, next is inulin. And one more thing, inulin. It is also a polysaccharide, but it is a homopolymer of. Till now we studied about homopolymers of glucose, homopolymer of N-acetyl glucosamine. This is a homopolymer of fructose. This is branched and this is unbranched. It is unbranched. Okay, unbranched homopolymer of fructose is what you call it as inulin. Okay, I hope that the class is not getting bored. Should I just revise it? No, or after the class I will be revising it. Okay, after revising only we will leave. So carbohydrates got over. Now next is amino acid. Carbohydrates got over. Now next is what? Amino acids. Now talking about amino acid. Amino acids are substituted methanes. Each amino acid is amphoteric due to the presence of basic amino group and acidic. Okay, so amino acids are, first thing itself is, they are amphoteric in nature. Do you know the reason why they are amphoteric? Because they show both basic and acidic nature, because this is the structure of this one, amino acid. See, COOH is present and NH2 is present. This shows acidic nature. COOH acid shows acidic nature and NH2 shows basic nature. So when both of them come together, it is a amphoteric carbon. And here you can have an alkyl group, and here you have hydrogen. So it is an amphoteric due to the presence of basic amino group NH2 and acidic carboxylic group. Amino acids contain an amino group and acidic group as substituents on the same carbon, that is alpha carbon. Hence they are known as alpha amino acids. So here you have your alpha carbon. Okay, this is your alpha carbon. In this alpha carbon, both of these are present. Okay, both your COH and NH2 is present on the same carbon, and that is your alpha carbon. So you can also call this amino acid as so call our amino acid as alpha amino acid. You can also call them as alpha amino acid. Okay, alpha amino acid. So they are amphoteric in nature, and the NH2 because both basic and acidic characters are shown by NH2 and COH respectively, and both of them are present on the same carbon, that we call them as alpha carbon. So you call this amino acid as alpha amino acid. Now do you know, when this alpha amino acids combine together, you form the protein. So what happened? So protein is the polymer of amino acids, or you can say amino acids are the monomer of proteins. I will make you do some questions. I have some previous year questions and some most probably asking questions with me. So for this Neet 2025, so I'll be just discussing about it also. Okay. Now amino acids. Amino acids. There are two main properties of amino acids. That is, the main property is it is their ionizable nature. Okay, we know that it is a Zwitter ionic form. Zwitter ionic form means, you know, this is your structure. Normal amino acidic structure is this one. Now Zwitter ionic form means NH3 plus COO minus H and R. This is your isoelectric. In an isoelectric medium, this shows this nature. Okay, in an acidic medium, this is shown in an isoelectric medium. Okay, means both are ionized. Both NH2 and COH are in the ionizable nature. Okay, in different pH, the structure of this amino acid changes. In isoelectric medium, this will be the structure. Means both of the acidic COH is and NH2 are ionized. One becomes COO minus, and another one becomes NH3 plus. In alkaline medium, you know, NH2 will be remaining as such. The acidic one will be changing. COO minus, R and H. This is an alkaline medium. That is, your alkaline substance will be remaining as such. The acidic substance will be changing. Okay. And then in acidic medium, just the opposite. The acidic medium will remain as such, and the one changing will be the basic substance, that is NH3. This is in acidic medium. So in different pH, they show different structures. That is their particular property. In different medium, they have different structures. That is their particular property. Okay, and this form is known as, this form is what you call it as Zwitter ionic form. Zwitter ionic form. You call this as Zwitter ionic form. Okay. Generally 20 proteinaceous amino acids present in the nature known as standard amino acid. Okay, now another properties, the chemical and physical properties of amino acid are essentially of the amino, carboxyl and R functional group. Okay, so their chemical and chemical nature or their physical nature, that depends upon this amino group, this acidic COH carboxyl group and the alkyl group, R group, R functional group. Not R functional group, not alkylic, I'm sorry. Now, classification of amino acid. Based on the number of carboxylic group, there are neutral, acidic and basic. If more number of basic substances are present in your amino acid, then it is known as basic amino acid. Neutral means same number of basic and functional, basic and acidic groups. Same number of basic and acidic groups, if same number of basic and acidic groups are present, then it is called as neutral amino acid. For example, you can see glycine. Glycine, you see the structure of glycine. The structure is this way: COOH, H, and H2C. One basic structure and one acidic structure. Neutral. Basic structure is not one, acidic structure is not, both are neutral. Here you have H. This is what? Glycine. This is the basic one, glycine. Then you can have an example of alanine. Alanine, the structure is somewhat same, only the difference is here instead of H, here it was H, in glycine here it was H, instead of H you can have CH3. Does CH3 come under any basic category or acidic category? No. So here also it is in a neutral way. Okay, this alanine structure is in a neutral way. So examples of neutral amino acids are glycine, alanine, and then you can have valine or you can have leucine. This in mostly, not mostly, some in are comes under this neutral amino acidic category. Now next is acidic. Acidic, it will have more number of acidic structure. For example, glutamic acid, the name itself suggests it, glutamic acid, aspartic acid. Okay, just remember the names. All these are acidic. Acidic, this one. So same amino acid. Now you can have basic amino acid. When you're talking about basic amino acid, lysine and arginine. Lysine and arginine. In neutral it was leucine. Okay, here it is lysine. Lysine and arginine. Then you can have some hydroxy amino acid. That is, they contain hydroxy group. See, serine. They contain hydroxy group. See, show you. That is a group containing OH. See, this is what you call it as serine. This is a hydroxyl amino acid. See, they are containing serine. They have an hydroxyl group. Okay. So, will you remember the different groupings based on the number of amino and carboxylic group? Yeah. So in neutral, same number of basic group and amino group acidic group are present, then it is known as neutral. For example, glycine. See, glycine structure you see, which is being here. In aniline structure, CH3. Other everything is different. Same. Only this thing is different. Valine and leucine are another example. Then acidic, the name itself suggests aspartic acid, glutamic acid. Okay. Basic lysine and arginine. Then you have some hydroxy containing group, that is serine. Then you can have some sulfur containing groups are like methionine. I don't want to study all these things, I am just giving the example. Then you can have some aromatic amino acids. Aromatic are also seen. Aromatic means cyclic structure. Okay, heterocyclic or homocyclic structures. Like example is tyrosine. There are cyclic structures. Okay. And tryptophan and all. Okay, they are aromatic amino acids. Phenylalanine. Okay, phenylalanine and all. They all are aromatic compounds, amino acids. Okay. So now we can move on to the polymer of amino acid, which is the who is the polymer of amino acid? It is mentioned it here. Somewhere mentioned, right? Protein is the polymer of your amino acid. Proteins. Proteins are organic biomolecules formed from C, H, O, N and S. Carbohydrates were formed from what all? Carbohydrates were formed from C, H and O. Here proteins are formed from C, H, O, H, O, N and S. All these together form your proteins. They are the most abundant and most diverse organic compounds. Okay, most abundant and most diverse organic compounds. Carbohydrates were the main energy providers. They are the cellulose was the most abundant polysaccharide. Okay, most abundant polysaccharide. The most abundant organic compound is your protein. Most abundant organic compound, that is your protein. Now in this itself, the most abundant protein in biosphere is rubisco. In plants you can see this protein, isn't it? Rubisco is the most abundant protein, MAP. But collagen is the most abundant protein in terms of animals. Animals means it is the most abundant animal protein you can see. Okay, the animal protein is collagen, but the most abundant protein, general category goes for rubisco. In animal category, the most abundant is collagen. Proteins are what? Macromolecules, and they are heteropolymers of amino acids. Okay, are they homopolymer? No, they are a heteropolymer, means different types of amino acids are joined together to make this protein. Synthesized in ribosomes. There's a 4 structure. Okay, now this proteins have 4 different forms, that is primary structure, secondary structure, tertiary structure and quaternary structure. Now primary structure means the linear chain structure, or you call it as polypeptide. Primary structure. Okay, it is polypeptide and it is the linear chain structure. The amino acids are connected together by peptide bond. For peptide bond, the first amino acid provides a COH group and the second one provides NH2 group. The left end of polypeptide is N-terminal and the right is C-terminal. Primary poly structure is unstable. This is an unstable structure, and you see that, see, like this it is. The first amino acid provides what? C. It is a heteropolymer, isn't it? So different types of masa can be seen. So C, just make it short. COOH. This is your first first amino acid. First amino acid. Then, and the second one provides what? NH2 group. Okay, they do not give CO group. NH2 group is given. This bond, see, one amino acid being connected to another amino acid. See, this is one amino acid and this is another amino acid. See this connection, this bond, can you see? This bond is what you call it as peptide bond. Here also you can see peptide bonds. Amino acid being connected to amino acid. These are peptide bond. Okay, so you understood what is peptide bond. 2 amino acids are connected together by peptide bond. Same way 2 glucose are connected to by glycosidic linkage. You have peptide linkage. Okay, and here the left end is N-terminal, and this is C-terminal, right end is C-terminal. Okay, just remember all these points. Left end is N-terminal and C, right end is CR. That is R stands for right, C stands for C-terminal, CR. Okay, so left end is N-terminal and right end is C-terminal. Okay, and this structure is unstable. Next you have secondary structure, isn't it? Secondary structure means it is more stable than the primary structure due to the presence of additional hydrogen bonds. You can see here additional hydrogen bonds can be seen here. Okay, and there are two types of this. These forms are between oxygen and hydrogen. We know hydrogen bonds are formed between oxygen and hydrogen. Now, two types can be seen, alpha helix and beta pleated. So, what is alpha helix? Like this will be alpha helix, here helical structure. Okay, this is your N terminal and this is your C terminal. Okay, and here you can see intra-hydrogen bonding. Intra-hydrogen bonding is seen. Okay, inside this you can see a hydrogen being bonded like this. See, intra-hydrogen bonding. Hydrogen is between the two acid. Okay, it is in a right-handed twisted way. It becomes an alpha helical protein. Example is your keratin. In hair and all, can you see your keratin? We see the keratin treatment and all in beauty parlors or in saloons and all. You can hear keratin treatment being done to hair and nails. This keratin is a alpha helix secondary structure. Next is beta pleated secondary protein. What is beta pleated? See, now like a pleats way. See, this is your beta pleated structure. Why do you call them as beta pleated? Means here you can see inter-hydrogen bonding. See, hydrogen bonding is between these two. Between, you can see between two layers you can see hydrogen being bonded. Okay, this is seen in keratin, in feathers and all. You can see feathers and in silk and all you can see keratin. This one is keratin in hair and nail. Okay, so I hope you understood what I meant by this. This is alpha like a helix structure and it is intra-hydrogen bonding between the molecules. You can see bond. N-terminal, C-terminal, left it is N-terminal, right is C-terminal. Intra-hydrogen bonding, keratin and beta pleated means inter-hydrogen. See, between here you can see bonding took place. Between the two layers you can see bonding taking place. It is inter-hydrogen bonding. Okay, now do you know what collagen has? This collagen has a triple helical structure. Okay, triple helical structure like this. This is how your collagen is seen. Okay. It was said by G. N. Ramachandran, Indian biologist. G. N. Ramachandran told that collagen has a triple helical structure. Now next is, I don't think that I will be able to take lipids in this class. I will be completing till proteins and I will be giving you rest. Okay. Okay. So tertiary structure. Next is tertiary structure. It is the maximum coiled form of a polypeptide. See, like this. You can see like this, like this, like this. This is your This is your tertiary structure in a maximum coiled way. This is your N terminal and this is your C terminal. Okay, here you can see hydrogen bond, ionic bond, disulfide bond, peptide bond, like that many bonds are seen in this tertiary structure. It is 5 bonds are seen: hydrogen bond, ionic bond, disulfide bond, peptide bond and hydrophobic bond. Many, so 5 bonds are there, therefore it is the most, sorry, not most, you can say the stable product. Primary is unstable. Okay, primary is unstable. Where is it? Yeah, this is unstable. This is a little more stable than primary. and tertiary is more stable than primary and secondary. Okay, because 5 bonds are there: hydrogen bond, ionic bond, disulfide bond, peptide bond, and hydrophobic bond. So 5 bonds are there, therefore they are the most stable. And peptide and disulfide bond are stronger than the others. So which are strongest? Peptide bond and which one? Next one, disulfide bond. So these 2 bonds are stronger than any other bond. Okay, all enzymes have this. Example is enzymes. All enzymes have this tertiary structure of protein. This is the most reactive form of protein. Enzymes and myoglobin and all have this structure. Okay, like this and all you can see this structure. See, sulfide, hydrogen bond, ionic bond. See, ions are all same. Ionic bond, and then hydrophobic bond, and then peptide bond. Peptide bond means between 2. Like this you can see peptide bond. Okay, now last is quaternary structure. This is the most stable structure of protein. More than one polypeptide present in this protein. Okay, for example, HB, hemoglobin. Okay, and all are example of this. Hemoglobin means it consists of 4 subunits are there in your hemoglobin. 4 subunits. In that, 2 of alpha type and 2 of beta type. Just remember this. In hemoglobin, 4 subunits are there. In that, 2 of alpha type and 2 of beta type. Okay. Okay. Now, one thing. Okay, and one more thing is denaturation. Denaturation means when high heat is given or when pH level is changed and all, the protein breaks. Protein breaks. And when it breaks, secondary, tertiary, quaternary, everything is broken, but primary remains intact. Primary is kept as it is not touched. Okay, denaturation means high heat is given or pH level is altered or electricity is given, anything. Such a case, the secondary, tertiary and quaternary breaks in terms of denaturation, but primary one remains intact. When you cut or when you break egg and albumin and all, it's broken. Same way. And all are examples. Okay. So primary, this one examples. Primary structure you don't have any example. Quaternary you have 2 alpha and beta. Alpha keratin in hair and nails. Beta is keratin in silk and everything, isn't it? Silk and feathers. Collagen has a triple helix structure. Okay, now tertiary means one more thing, that is the secondary has in secondary alpha helix has intra hydrogen bonding, and this beta pleated has interhydrogen bonding. Then tertiary structures are there. Tertiary means continuous or maximum coiled form of polypeptide. It is stable than the other two because of 5 bonds. It has hydrogen bond, ionic bond, disulfide bond, hydrophobic bond and peptide bond. In that, disulfide and peptide bond are the strongest. Example, in most enzymes, every enzyme, not most, all enzymes are in the tertiary structure. Okay, enzymes and myoglobin are all examples of tertiary structure. And quaternary you can have is the most stable structure, HB. Example is your human hemoglobin. 4 subunits are seen, 2 alpha and 2 beta. Now, types of protein. On the basis of their constitution, proteins are classified into 2 main groups, that is simple proteins and conjugated proteins. The protein contains only amino acid as simple proteins. Conjugated proteins means it has amino acid and non-protein groups. Okay, same way as homopolymer and heteropolymer. We know that protein is a heteropolymer of amino acid. Okay, but not same way, there is difference, that is this simple means only amino acid, not one type of amino acid. One type of amino acid means homopolymer, but proteins are heteropolymer, 2-3 types of amino acid combined together. Simple protein means only amino acid is constituted in it. And conjugated means mixed is the meaning, isn't it? Conjugated means amino acid and some non-protein groups, are also seen. Okay, and some protein and their functions. That is another topic. So you see, next is the last topic for part 1. There are some proteins and their functions. So you see, the protein is collagen, and it forms what the function is. It is an intracellular ground substance, isn't it? Collagen, not glycogen. Okay, collagen. Collagen means it is the most abundant animal protein. We have studied it. So it forms intracellular ground substance. Trypsin is an enzyme. Insulin is a hormone. Antibody, we know, fight infectious agents. Receptor, it's sensory reception, smell, taste, hormone, etc. Okay, receptor receives. Okay, sensory receptions of smell, taste, and etc. And GLUT4, it is enables glucose transport into cell. This is very important. Questions are every time asked. GLUT4 enables glucose transport into cells. Not this one, okay. This whole table is important. Collagen forms, it is the most abundant animal protein, 1 point. First point. Second point, triple helical structure is therefore helical. So it is alpha helix structure, isn't it? Triple helical structure is therefore collagen. And third is, it functions as intracellular ground substance. Trypsin is an enzyme. Insulin is a hormone. Antibody fights infectious agents. Receptor, it is a sensory reception to receive smell, taste, etc. hormone, etc. GLUT4 means transport or enables glucose transport into cells. So this is the thing. Now should I continue with lipids? Let me just check. Lipids, glycerol you have to learn, fatty acids. No, that I will just continue in part 2. Part 2 will be uploaded by today evening if time permits, or else by tomorrow morning. Tomorrow morning by 12 to 1 it will be uploaded. Okay. So this is the things which we learned today. We learned about biomolecules, which is present in protoplasm, that is cytoplasm plus nucleus. Don't go anywhere, I will be discussing the questions. Cellular pool, biomolecules form a cellular pool in protoplasm. 40 biomolecules are present, almost 40. It is metabolite because it helps in metabolic activities. Then CONH are the most, some of the most seen elements in our nature. It goes in the order oxygen, carbon dioxide, nitrogen, hydrogen. Oxygen is the most abundant, whether in Earth's crust or in human body. Okay, in Earth's crust the order goes as OHN. So OHC and N. Okay, now biomolecules can be differentiated with the help of 2 tests, that is ASH test and acid filter test. ASH test helps the biomolecules to be differentiated into organic and inorganic. Acid filter test into micro and macro. Now you know the difference of micro and macro, I hope. Macro molecules which weigh less than 1000 Dalton, micro which weigh more than 10,000 Dalton, macro. Okay, micro is filtrate, macro is retentate. Lipid is an different example. Now I have to teach lipid. I will start from lipid in the second part. So then I will tell you the reason why lipid is considered as retentate, even though it is a micromolecule. Its size is less than 800 Dalton, but it is considered as a retentate. That is, they are acid insoluble. Then you have to know that water, 70 to 90% in our cell is composed of water. Protein 10 to 15%, carbohydrate 3%, lipids 2%, other inorganic ions 1%, and your nucleotides, that is RNA, DNA and all. RNA is not there, but still all it is of 5 to 7%. Now metabolites are of 2 types, primary metabolites and secondary metabolites. Metabolites which help in growth and development are primary, which do not help in growth and development but are only used for human welfare are secondary. Examples for primary, amino acid, lipids, and for secondary it is, examples are rubber, gum, concuvalin A, etc. Many examples are there, and they are absent in animals. They are present in plants only. Okay, we get them from plants. They do not do any function of metabolic for the producer. Now carbohydrates are the first one which we studied, first biomolecule. Carbohydrates, there are different types: monosaccharide, oligosaccharide, and polysaccharide. Mono means only, they cannot be hydrolyzed further, they cannot be hydrolyzed. Oligo means they can be hydrolyzed. Oligo are also differentiated into 3 categories: disaccharide, trisaccharide, tetrasaccharide. The disaccharide means when disaccharides are hydrolyzed, you can get 2 units of monosaccharides. Okay, examples of monosaccharides are glucose, fructose, ribose, ribulose, glyceraldehyde, etc. Then disaccharides example is maltose, sucrose and lactose. Maltose means 2 units of glucose, alpha 1,4 linkage. Sucrose means 1 unit of glucose and 1 unit of fructose, and lactose means 1 unit of glucose and 1 unit of galactose. Okay, glycosidic linkage or glycosidic bond is same. Now sucrose is a non-reducing sugar, therefore in plants carbohydrates is mainly transported in the form of sucrose. Now glucose example is glucose is also called as blood sugar, or you can also call it as grape sugar. It is hexose aldose and it is a reducing sugar. The habit structure is I have shown there. Fructose, hexose, ketose, reducing sugar, fruit sugars, and it is also seen in seminal plasma in male. Then as polysaccharides, the first example I taught you was glycogen. It is a branched homopolymer. It is a storage polysaccharide, and you know the right end and left end has different names. The right end is what you call it as reducing end, and the left end is non-reducing end. And there are two types of polysaccharides. There are macromolecule and they are homopolymer or heteropolymer. Homopolymer means only made up of one type of monosaccharide. Heteropolymer means made of different types of polysaccharides, monosaccharides. Okay, heteropolysaccharide example is hemicellulose. Now in different examples I taught you for polysaccharide. Cellulose, it is a homopolymer, unbranched homopolymer. Glycogen is a branched homopolymer. Chitin is a unbranched homopolymer of N-glucosamine, N-acetyl glucosamine, and inulin is a homopolymer of fructose, and starch homopolymer of glucose. The starch contains 2 units, that is amylose and amylopectin. Amylose is unbranched and amylopectin is branched. Okay, the amylose is the one which converts iodine into blue color, and amylopectin converts as a branched one. Okay, it doesn't convert anything. Okay, fine. Now amino acids, I think you know amphoteric is there, a neutral. It is an amphoteric. Amino acid is amphoteric, but it shows different structure in different medium. That is, in isoelectric medium this structure is shown, in alkaline medium this structure is shown, and in acidic medium this structure is shown. Different medium it shows different structure. Then neutral, same number of basic and acidic groups, glycine, valine, alanine, leucine and all. Then acidic means aspartic acid, gluten. More number of acidic group is shown. Basic means more number of basic group. Examples are lysine and arginine. Then hydroxy, it contains hydroxy group. Serine is an example. Aromatic tryptophan, heterocyclic aromatic compounds, tryptophan, tyrosine, phenylalanine, etc. Then proteins, that contains CHON and S, that is the most abundant organic compound. Rubisco is the most abundant protein. Most abundant animal protein is glycogen. Important points, these are all important points. Okay, now heteropolymer of amino acids, they are heteropolymers. Okay, it is made up of more than one type of amino acid. Now proteins, primary, secondary, tertiary, quaternary. Primary you know, secondary also I told you, tertiary and this one. And proteins are of simple and conjugated. Simple means amino acid, made up of only amino acid. Conjugated means amino acid plus some non-protein substances also say. Now collagen, intracellular ground substance, trypsin enzyme, insulin hormone, antibody, fight infectious agents, receptor, sensory reception, and GLUT4 enables glucose transport into cell. Okay, so now I will be discussing some PYQs and some possible questions, and I won't be uploading it. So listen carefully if you want. Listen carefully if you want this, okay? I won't be uploading. If I uploaded it, then if I am showing it in the slide, then you will be looking into it and won't be listening the questions. So I'm just directly showing it. Because I'm not showing it directly. I'll be just reading it. So just listen. Let me just take the page 104. 104. Okay, so first question is, first it said they asked about lipids. Okay, so first question, identify the incorrect pair: drugs ricin, alkaloids codeine, toxin abrin, and lectins concavalin A. This I taught when we talked about secondary metabolites. So lectins concavalin is correct, toxin abrin is correct. Yeah, the wrong one is drugs ricin is wrong. Drugs is vinblastine, curcumin, risperidone. Ricin is a Toxin. Resin is not a drug. So it is first option, drug, resin. Second question, which of the following are not secondary metabolites in plants? Rubber, gums, morphine, codeine, amino acid, glucose, and last option, vinblastine, curcumin. It is amino acid, glucose, and sorry. Which of the following are statements with reference to lipids and lipids? No need. Concavalin A is an alkaloid, an essential oil, a lectin, a pigment. It is a lectin. Which of the following are not polymeric? Proteins, polysaccharides, lipids, nucleic acid. It is lipids. Lipids are not polymeric, they are monomeric. Okay. Now, the question, it was question number 1, 2, 3, 4. Okay. Fourth one. Now, next question is, let me just check, okay. I can't see anything from which we learned. This previous year questions I am asking. Which of the following is the simplest amino acid? Yes, simplest amino acid is, I taught you. Simplest amino acid is glycine, isn't it? HH. Coming, see, simplest amino acid. What is the structure of glycine? See, HH. Simplest amino acid. Now, a protein is imagined as a line. The left, this is assertion, okay? The left end represented by first amino acid, C terminal, and the right end represented by last and N terminal. Wrong. C is right terminal and that itself is wrong. Adult human hemoglobin consists of 4 subunits, 2 subunits of alpha type and 2 subunits of beta type. It's correct. So statement 1 is false and statement 2 is true. Okay. 17th one, option 3. Okay. Now, which of the following is the most abundant protein in animals? It is, of course, collagen. Ramachandran plot is used to confirm the structure of which one? DNA, RNA, proteins and triglycerides. Proteins, because he was the one who confirmed the structure of collagen. Which of the following glucose transporters is insulin dependent? Which is insulin dependent? GLUT4, isn't it? GLUT4 is the only example we studied, so just remember that GLUT4. Okay. The amino acid tryptophan is the precursor of the synthesis. So that is an old question. It won't be asked, so no need. Even though the answer is melatonin and serotonin. Okay. Which of the most abundant protein in the animal world? Collagen. Same question. Cellulose does not form blue color with iodine because cellulose. Last question. Which one of the following is a non-reducing carbohydrate? Maltose, sucrose, lactose, ribose, 5-phosphate. It is sucrose. And one more question. Is there one more question? No. One more question. Cellulose does not form blue color with iodine because it is a helical molecule. It does not contain complex helices and hence cannot halt iodine molecule. It breaks down when iodine reacts with it and it is a disaccharide. So which is the option? It does not contain complex helices and hence it cannot hold iodine molecules. The complex helices structure, which is starch, it holds. In starch also amylose holds iodine structure. Okay, so that's all for the part 1 video. I will be uploading part 2 most probably tomorrow morning itself, because already when I'm uploading it's already evening, so most probably by tomorrow morning I will be uploading the part 2 video, and by tomorrow I will be completing the chapter. We will be doing more questions. Today I only got to do some questions because it's already more than 1.5 hours. Okay, so thank you. See you all in the next video. Bye.